I would like to introduce myself. I'm Olivia Stricker. I'm with the DiaPharma group and the host of today's webinar about net biomarkers. For those of you that are familiar with DiaPharma, you know that we specialize in distributing consumable products that are specifically related to everything hemostasis. And our interest in hemostasis and coagulation has led us to focus energy on NETosis and biomarkers in that space. And also a relationship with VolitionRx to distribute their Nu.Q H3.1 discovery research use only ELISA kit in the US and Canada. So VolitionRx is a multinational epigenetics company that's powered by Nu.Q, its proprietary nucleosome quantification platform. Volition is developing and commercializing simple, easy to use, cost-effective blood tests with the hope of being able to help diagnose and monitor a range of diseases, including cancer, some cancers, and NETosis-driven diseases such as sepsis. I have Dr. Terry Kelly with me today, who's going to give the presentation about net biomarkers. Dr. Kelly is the Chief Innovation Officer for VolitionRx and also the Chief Scientific Officer of Volition America. She has more than 15 years of experience related to epigenetics, including developing novel technologies and seeing them all the way through commercialization. Her laboratory is based in San Diego, where she uses a multi-omics approach to understand really the basic mechanisms of epigenetics. She's prepared a 20-minute presentation that will be followed by a question and answer session. For anybody here, please feel free to use the Q&A tab at the bottom to submit questions, and I will kind of organize them and march us through those at the end. But please feel free to type them as she's going. And then to start, I've put up this 2-question poll about what your, most often go-to assays are for NETosis and what, your lab is using as a positive control. So I don't know if everybody can see them. Let's see, I will give a couple more, a little bit more time for the poll, and then looks like people are still answering. But it looks like citrulinated histones is really popular. And let's see, coming in second is, I think, probably a double-stranded DNA assay. And let's see. Also, people are using PMA and ionomycin stimulated neutrophils as their positive controls. So I think that that's super interesting. And with that, I would like to welcome everybody and hand it over to Terry. So let me stop sharing this and all right.
Great. Thank you so much, Olivia, and thank you everybody for joining today. So today I'm going to talk about NETs, and as Olivia mentioned, one of the assays that we've developed specifically looking at H3.1 nucleosomes in clinical samples to measure NETosis. So as I'm sure many of you know, there's multiple different layers of immune defense mechanisms. The first is obviously at the entry point, and so having things like your skin and mucous membranes to prevent any pathogens from entering the systems. However, once that is broken and you actually have pathogens that have entered the body, we have a variety of different immune cells that will respond to those pathogens. The first is this innate immune system, where it is really responding to acute infection, which ultimately transitions into the adaptive immune system with long-term immune responses. And so today, as many of you know, we're going to focus on neutrophils. So neutrophils are the most abundant white blood cells in humans, making up over 50% of the circulating white blood cells in the blood. It is part of the innate immune system. And then interestingly, so in response to pathogens, they rapidly migrate to the sites of infection and inflammation, where they will phagocytose, neutralize, and eliminate invading pathogens. And they do this by the series of receptors that are on the outside of the neutrophil that are really able to sense a variety of different pathogens that are in the environment. Once they do recognize those pathogens, they'll migrate to the site of infection, and they will essentially release their chromatin extrusion into the circulation and basically capture a lot of those pathogens. This process of NETosis was really only recently described in 2004. And importantly, and what we'll come back to, is that these nets contain these microbial granular enzymes, neutrophil elastase and myeloperoxidase, in addition to histone proteins and nucleosomes. And so while ultimately you end up with this extrusion of chromatin, there are a couple of different mechanisms for NETosis to occur by which. So the first is suicidal NETosis, and this is in which the cell will actually die. The neutrophil doesn't survive this, and all of the chromatin is extruded out into circulation. There's also a second form, which is called vital NETosis. And so in this case, the cell is actually able to survive, and only a portion of its chromatin is extruded out, but the cell is able to continue on in function. And so while these different mechanisms are interesting, and it's something my lab here on the innovation side is really spending a lot of time to understand the different triggers and the regulations between these two mechanisms, for the purposes today is really that at the end point for both of these, there is an extrusion of the DNA, the chromatin, and these microbial proteins into circulation. And so while the pathways by which the NETosis is occurring is different, in the end point, they're still making those nets. And this is exemplified here in this cartoon on the left, where you have the intact neutrophil, where it extrudes the chromatin. And you can see here that those chromatin fibers have both the nucleosomes made of this octamer core. In that octamer and each of those histones, they have a tail. And as you guys all know, if you're using the citrulline-based assay, you have here is a histone tail that has the various residues that have been citrulinated. This figure on the right here is an EM figure, and I think it just really nicely shows where you have the neutrophil itself, and now here you have the net that's been extruded out. You have the purple here is the bacteria that really shows the net being able to capture some of those pathogens, but importantly also you have that it is capturing red blood cells, and so these nets can be quite large. They can be up to 15 microns in diameter. And so with that, it can capture not just pathogens, but some of the surrounding cells as well. So as a result of that, the nets, while they're really important for the innate immune system and protecting us, it can also have detrimental effects if the nets is not, if NETosis goes unchecked, and it can become pathogenic. And so some of the examples of that is that it can cause damage to other cells that are around. It can activate macrophages and interact with macrophages to increase inflammation. This picture in the middle is really showing how it can capture red blood cells and other circulating cells to essentially cause occlusion and ultimately thrombosis. There's also a lot of interesting literature about how nets and neutrophils interact with tumor cells and play a role in cancer progression and metastases, as well as I'm sure many of you know about its role in autoantibody generation with things like rheumatoid arthritis and lupus. So with the importance of NETosis, both on the innate immune system, but also the importance for the role it can play in pathogenesis, there's been a variety of groups that are interested in studying it and a variety of methods that have been used. So I'm just going to touch on a little bit of them today. I'm sure most of you guys are familiar with all of these. And I think I just really like to highlight that, because of nets, their complicated structure, their importance to disease and potential pathophysiology, and NETosis is really a dynamic mechanism, is really understanding what markers they have, what's preceding NETosis, and what's during the resolution of nets often needs different biomarkers depending on the question. And so my goal here isn't to say that there is some markers that are better than others, but it's really more about choosing the right marker and the right assay for your needs. So I'll start off with one of the more commonly used, and so this is myeloperoxidase. So this is one of the antimicrobial proteins that is present in the nets. And so this is often measured by itself, or as many know, in combination with anti-DNA antibodies or fluorescent DNA intercalating dyes. So my MPO is used both to measure by itself and then in combination with DNA. Similarly, as neutrophil elastase, I think is used a little bit less commonly than MPO, but it can be measured by itself or also with DNA. The citrullination, which many are looking at, I think one of the things that's really nice with citrullination is that it plays a functional role. And so once the H3 is citrulinated, it plays part of the chromatin decondensation pathway. And so for me as a basic scientist, anytime there's a mechanism involved, it gives me confidence to the importance of a biomarker. So the other part to know about this is that it really is specific to PAD4 mediated NETosis. And so it's not, as I showed in the beginning, there's different mechanisms for NETosis. The PAD4 mediated citrullination is really focusing on that kind of suicidal NETosis. Also, as I mentioned, the nets is a very dynamic situation. And so some of these histone tails are really prone to cleavage. Some of those cleavage sites are upstream of some of the citrullination sites. So depending on what residue you're looking for, you may either not see it because that histone tail has been clipped off, or you may see it disappearing. And it's not because it's a loss of the citrullination per se, it's really just that loss of the histone tail altogether. And then lastly is using cell-free DNA. And so this is a core component of the nets and neutrophils, but I think it's also important to know that cell-free DNA is not specific to nets. It is released by any cells that are dying into circulation. So looking at those different biomarkers, there's a variety of different assays that are used. From very straightforward and easy to more complex. So the first is microscopy. And so in many ways, a picture is worth 1,000 words. And I myself like to really actually see pictures of the nets. But one of the challenges with that is it can be very time consuming to do the staining. It does require specific microscopes in order to do the immunostaining. And care also has to be taken with fixation to make sure that you're not over fixing the cells, which can induce NETosis in and of itself. The other question is with quantitation. And so there's, depending on how you're analyzing the data, if it's just by eye, then it's really subjective in terms of quantification. However, there are software packages that can be used to do more accurate objective quantification. Similarly, there's flow cytometry out there and that people are using, and this is looking at cell surface markers, but it's also able to measure the size of the neutrophil prior to release, and along with some differences in side scatter to measure NETosis. As with microscopy, you want to be really careful with preparing your samples for flow cytometry, and these require some specialized equipment using fax machines. However, it is very quantitative, and so that's what some of the benefits of it. Next, DNA quantification. So there's a variety of different tools that can be used to quantify DNA, from spectrophotometers to qubits to picogreen. So doing DNA quantification is very sensitive. It's objective. You get an absolute number. However, as I mentioned, it's not specific. And so I think really being careful about the conclusions that you make and the experimental conditions that you've set up if you're going to use DNA quantification as the main readout for NETosis. Lastly is ELISAs, and so this is one that's become quite popular. One of the things that's beneficial with ELISAs is it's very objective. You do get a number as a readout. It can be quantitative. There's questions about specificity, and this really goes to the assay development and the antibodies that you're using, the controls that you're using, the blockers that you're using. So I think that's just where a lot of care needs to be taken in developing ELISAs, specifically for looking at plasma samples, because there's a lot of additional challenges that come up working in clinical plasma samples that you don't see in cell culture lysates. Along with that specificity is then reproducibility. And are you able to get the same results over time if you are making a new batch, down from the antibody lot to all of the various buffers as well. And lastly, for ELISAs and generally for everything is still standardization. And so in the field, there's still not one sort of global standard that's used for measuring NETosis. And so this is something that we in many groups are working towards. So what our approach is for measuring NETosis is really focusing on the nucleosome. So for those of you that don't know, a nucleosome is comprised of a histone core, which is an octamer of duplicates of those 4 histone proteins. Around that histone core is about 150 base pairs of DNA. Nucleosomes are relatively stable, and this is because of those interactions between that protein octamer histone core and the DNA. So relative to other analytes that are in circulation, the nucleosomes do stay around for a fair bit of time. It's obviously a core component of NETs, which is important, and they contain a variety of modifications to both the DNA and the histone core. And so this will allow us to look at things like citrullination. But as I mentioned, as we look at different NETosis mechanisms, maybe there will be some other histone modifications that will become relevant and that are part of that decondensing of chromatin ahead of NETosis. So this is what our assay looks like, the H3.1 Nu.Q assay. We use a capture antibody, which is directed towards the H3.1 histone, and that will bind to the histone. And then we have a label detection antibody. That label detection antibody is actually recognized as a conformational epitope, so that this is how we know that we're really measuring nucleosomes and we're not measuring any free histones or free octamers, is that antibody will not bind if DNA is not also present. Couple of the benefits of the Nu.Q assay is that it is objective. You get a concentration of nucleosomes is the readout, making it also quantitative. Over the years, we have worked with a variety of different input materials, all the way from cell culture to plasma. So we know that it's robust across a variety of different matrices. And importantly, we also developed this with clinically oriented questions in mind. And so knowing that we ultimately wanted this assay to work in clinical samples, we spent a lot of time developing it, making sure that it performed the way that clinical assays needed to. So focusing on things like linearity, sensitivity, precision, and interference. And so I'm going to show a little bit of that data today. And so this is looking at our linearity data. And so the way that we did this was we took 2 different samples, a sample that had very low levels of nucleosomes and one that had very high levels of nucleosomes. And we then mixed them at different ratios. And then we measured Nu.Q across that range. And so here's the results from 3 different lots. And so our overall linearity was 89% reproducibility across those lots, because we want to make sure that if you get a result today and do the same assay in a year from now, you're going to get the same result. Next was really, when it comes to clinical assays, is making sure we are able to interpret that quantitative results that we get. And so we did a series of studies across all of the 3 different lots in this case at this point, looking at things like the limit of blank. And so this is running a variety of samples that are not expected to have the analyte, and to really assess what that background level is. Also the limit of detection, to know how low we are able to go with this assay in particular. This typically isn't an issue with NETosis related diseases because nucleosomes and NETs are so elevated. But we really wanted to focus on those early stages of NETosis. So we wanted to make sure that it was able to be as sensitive as possible. Next is limit of quantitation. And so when you get an actual number, that's read out, are you able to believe those differences? And so this for us is 22.7 nanograms per mil. And so if we have quantitative differences above that 22 nanograms per mil, then we know that that is a real quantitative difference. Next, we looked at precision. And so in this example here on the left is looking within run. And so we took, in this case, 4 different samples that spanned the range of the Nu.Q assay, and we ran them for a minimum of 10 days over 2 operators. And you can see that the consistency within a run was quite good. And also, even more importantly, is across the runs, we were able to get good consistency, speaking to the reproducibility and reliability. Next, we looked at something that really is specific to plasma samples and clinical samples, and that's interference. And so we know that there's a lot of other things happening in circulation and that can interfere with assays. And so we really wanted to understand is what are the limitations of the Nu.Q assay in terms of those common interferences. So the one that I've highlighted here that we see a lot of in the lab is just hemolysis. And so here you can see that the Nu.Q assay still gives reliable results, even up to 500 milligrams per deciliter of blood. So now that we have all of the data showing the value of the assay and the the robustness of it, what about the utility? And so these are a paper on the left and an abstract on the right, really looking into nucleosome levels. This was our first foray into sort of NETosis, and it really came about with COVID. And so on the left, this was looking at COVID positive patients that were in outpatients or the normal ward, separated also then with those that were in the ICU that survived and died. And so you can see that the nucleosome levels were most elevated in the patients that had the worst prognosis. We then expanded beyond that and looked in a new cohort. And on the top on the right is we separated out patients that were non-severe versus severe. Severe in this case means that they're on organ support. And we can see that correlation that those majority of the patients that were on organ support had much elevated Nu.Q levels. On the bottom here is now looking at, so okay, nucleosomes are elevated, what does that mean? And so in this study here, we were able to show that the patients that were admitted into the ITU, when you look at nucleosome levels and correlated it with survival at 28 days, we saw that the patients that had the highest Nu.Q levels had the poorest outcome for survival at 28 days. So looking in clinical cohorts about how Nu.Q performs compared to other markers of NETosis is what we're showing here. And we can show that it performs pretty similarly. And that we can look on the left and we see different levels between control patients and those with sepsis, and also between those controls and COVID, and also sepsis and COVID. So this is consistent with what some of the other assays are able to show. And I think just really speaking to using a variety of different assays to really give you confidence that you are measuring NETosis. So in summary, NETs are an important part of the immune system, and dysregulation can lead to pathology. There's a variety of ways to measure NETs, each with their own benefits and limitations. And our H3.1 Nu.Q assay is ready to use. There's no customization or optimization required. The kit comes with everything you would need to run the assay. I think importantly, we've developed it to work with clinical samples and now have sort of released it for RUO use as well, but you still get all of the benefits, if you are working with clinical samples, and it also allows you to use the same assay all the way from if you're studying something in cell culture and in cell lysates all the way to clinical samples. It gives you the utility of using that same assay all the way through. And then lastly, to look at utility and clinical relevance, as we show that it does correlate, Nu.Q levels do correlate with risk in both COVID and sepsis. And so lastly, this is the work of a variety of different teams. So the team on the top, this is our group in Belgium, which is where all of our product development and production happens. So we do everything from making the antibodies in-house to making the standards in-house to all of the buffers. And so this is the team both on the R&D production and quality sides that lead to the assay. And then the team on the bottom, this is my team based out here in San Diego, where we're using Nu.Q and other methods to really go one step further to understand what are the downstream mechanisms downstream of after a net is released, and also upstream as well. And so with that, I would like to thank everyone for this attention and open to questions.
Fantastic. Thank you so much. That was a really great overview of the options out there, and kind of a little deeper step into the Nu.Q Discovery 3.1 kit. So some questions have come in, and I will kind of start broad and maybe work narrow. So one of the first and more common questions that I've seen is, this isn't necessarily a super specific marker for NETosis. So can you talk to how measuring H3.1 is a NETosis marker generally?
Yeah, absolutely. So as I mentioned, anytime cells die, they'll release nucleosomes into circulation. Nucleosomes themselves can be fairly toxic to cells, especially when they're separated, the DNA and the histones. So we have really good clearance mechanisms. So they're continually cleared, for example, by the kidney. And so really, when we think about measuring nucleosomes, we think about measuring this imbalance between how quickly are they released and how quickly are they cleared. And so you'll see nucleosome elevations, for example, with extreme exercise. So people that run marathons, you can have increases in nucleosomes, but they tend to resolve very, very quickly over time. What happens with something like NETosis, it's really just the magnitude. And so you'll see it's a lot more nucleosomes that are being released. And so for some of you that may know that we also are measuring nucleosome levels in cancer, we'll get nucleosomes that are elevated that are in like the 60 nanogram to 100 nanograms per range. When we're looking at some of these NETosis related diseases, it's a much higher elevation of nucleosomes. So I think that's one part of it. What we also do is we, in a lot of these samples, we'll go and do sequencing afterwards, and we can look at the DNA sequence, specifically looking at methylation, and confirm using a cell of origin analysis that the DNA that we're looking at is actually coming from neutrophils as opposed to other cell types.
Okay, and another question that we get quite frequently is, does this measure free H3 histones, and is there a way to measure free H3 histones?
Yeah. So our specific assay does not, and that's by design. And so this is because of that detection antibody that it really does require the DNA and the histone octamer to be there. So we've done a lot of development where we will add free histones by themselves, we'll add free octamer, and we show that we won't get any signal without the DNA actually present in those samples. So we know that our assay is specific to nucleosomes. Could you do free histones? Absolutely. I think that what you need to be mindful of and careful of is which antibodies you're using, right? Because you don't want to have the same antibody to the same epitope, because you'll have steric interference, potentially not enabling them both to bind. And so I think there's a lot more that you would need to make sure that you're looking at, that you're able to interpret your signal.
Okay. Another question about citrullination. Somebody's asking if cell types other than neutrophils produce citrulinated H3.
Not that I'm aware of. I think it really is very predominantly associated with, at least with PAD4. So perhaps if there's other cells that have PAD4 activation and downstream potentially, but I do believe that it is mostly associated with NETosis.
Okay. And then curious, what are the hurdles that people are facing when they're trying to, establish their biomarkers in cell culture and then transfer them to clinical interpretation? Like, what are the biggest obstacles?
So I think the biggest obstacles are really the complexity of the matrix and the abundance of your analyte. And so I think, when you get into blood, there's albumin, there's a lot of sticky things in there. So I think there's high levels of albumin. There's other antibodies potentially that are circulating that can interfere, that can have bridging effects. And so it's just that complexity of the matrix. And so really making sure that you have all of the appropriate blockers and all the appropriate controls to prevent any non-specific binding from happening.
Okay. And that is the end. Let's see. Let me see if any more popped up while we were chatting. Oh, yes. Can you measure NETs in whole blood, plasma, serum? Can you speak to the serum? Because I know that that's a...
Yeah, so people do, and we started working with serum. So yes, you can. However, it's much less of a forgiving matrix in our hands. And so when, again, speaking to wanting to develop something that was robust and reliable and could work broadly, we found that with serum, you had to be much more careful about when you processed it and how long you processed it. And we just didn't find that it was as stable. Nucleosomes were not as stable of an analyte in serum compared to plasma. And so that's where we ultimately recommend using plasma. It is a lot more forgiving in terms of having nucleosomes be stable, being able to have a little bit more flexibility in your processing.
Okay. We have another question. Can this kit also recognize eosinophil extracellular traps? It says, if so, how can we estimate the levels of NETs?
So
Not really
yes.
NETs, they're just extracellular.
Yes, so we are, again, the assay itself is fairly agnostic as to where the nucleosomes are coming from. So if they are, you have eosinophil extracellular traps, absolutely, that we would be able to recognize them in that case too. I think if it's important to differentiate between the two, I think that's where you would use this as the first step, and then you would reflex to some other tests to then really confirm, okay, where are those NETs actually coming from?
Okay.
And so that's something, if you're interested in doing, we could talk more about that as well.
Is this human specific?
So no, it's not. And so that's one of the things that's very nice about histones and nucleosomes is they're conserved across species, all the way from yeast into humans. So we have a different branch of our business where we're actually looking at circulating nucleosomes in dogs. So we know that the assay works as it is in canines. We've done other research-based project looking at other species as well. But that is one of the values of this. I'll also add that with the ELISA, it works on 20 microliters of plasma. And so we wanted to be able to have something that could be used for mouse models, knowing that it's very difficult to get a lot of material from mice. The input volume for this is at 20 microliters.
Okay. There's tons of questions. Healthy versus sepsis patients, sort of like the general value estimates.
Of nucleosome levels?
Mm-hmm.
So the nucleosome levels in healthy patients, they tend to be 50 nanograms per mil or less. And so then you'll get sort of up to, depending, it's very, then there's a whole bunch of caveats of what you're going to have in terms of, between stages and disease states and everything. So really it's normal is less than 50, and then elevated is, the more elevated, the more sick, essentially, which is what we showed with some of the sepsis and COVID, that they can be elevated in sepsis, and in COVID, but then the ones that are going to actually have the worst prognosis are even further elevated.
Okay. So we have officially run out of time. I didn't run out of the questions to ask, so I will get a list of the questions put together that we addressed and that we didn't address, and they will be in the follow-up email. For anyone that is wanting more information from DiaPharma, we distribute the nucleosome H3.1 Nu.Q ELISA kit in the US and Canada. So please reach out. You'll get like a survey to this webinar following the end of the webinar, and it will also be available on demand. So we very much appreciate everybody's time and attention, and hope you enjoy your probably frigid day, I would say, because it's freezing everywhere in the US, probably but San Diego. Yeah. And yes, thank you so much, Terry.
Thank you, everyone.
Okay, bye guys.